Thank you very much. It's an absolute pleasure to be here this morning in Milan. And for me, it's particularly a joy because I just, as I was saying last night, had completed reading the Isaacson biography of Leonardo da Vinci. So I had a great chance yesterday to see some important sites where his magical genius had taken place here in Milan. And then tonight I will fulfill a lifelong dream and attend a performance of Simona Boccanegra at La Scala. So I'm delighted to be here, but I'm especially delighted to be in Milan. That said, my presentation is a little bit about Foundation Medicine with an emphasis on this first part in lung cancer. And we've all know that cancer is a disease of the genome, and this isn't an audience that needs any reminders about whether the germline predisposition is contributing a major or a minor part, or the environmental exposure to cigarette smoking or ultraviolet light or radiation or something like that is contributing the major part. Either way, the patient has cancer, and we want to use precision medicine to get them a better outcome than we do when we use one-size-fits-all treatment. And when we introduced this approach with metastatic melanoma driven by BRAF V600E base substitution, targeted with the first anti-BRAF drug, vemurafenib, we saw this kind of change in a bone scan so quickly after the targeted therapy had been introduced, and that gave us great excitement that this was going to make a major difference in final outcome of cancer. Well, cancer genomes, as we all know, are very complex, but although the genome of the human has maybe 30,000+ alterations, the cancer genome alterations are not in the tens of thousands. They're more in the hundreds or even less. And we also know, of course, that children have very few alterations in their malignant processes. They haven't been exposed to cancer-causing environmental agents like adults have. But adults, particularly cigarette smokers or sun worshippers or whatever, have exposures that give them complex cancer genomes. But when you really look at it, the biologically relevant genomes in any one individual's cancer, when you do DNA sequencing, is generally relatively small, with only maybe 5 to 10 biologically significant genes per patient, and really only 1 or 2 important clinical drivers. Now, in some patients, that number can be a lot higher, and unfortunately, in a fair number of patients, the actual drivers are very small in number and not targetable. The history of the application, I can personally mention that this December in 2018, we'll celebrate the 20th anniversary of the true birth of this approach to cancer. And that was a day in December of 1998, kind of a dreary, dark, snowy, rainy day in Bethesda, Maryland in the US, when on one side of the street at the US FDA, the anti-HER2 cancer treatment drug for metastatic breast cancer, Herceptin, was approved, and on the opposite side of the street, an immunohistochemistry test, the DACO Hercept test, was approved, with each mentioning the other in the label, the birth of the era of companion diagnostics. And since then, of course, we've had quite a number of them, and I'm going to talk about the recent approval of Foundation Medicine's FoundationOne CDx, which actually summarizes all of the approved companion diagnostics in one complex DNA sequencing assay. I've had a chance to look at all of these. It's only up through 2016. There have been several in 17, and already 2018 is yielding new ones. There's been an enormous list of drugs and therapies that have gotten into development now. And this is, of course, a multi-billion-dollar industry, and it's growing at an exponential rate. Clearly, for cancer patients, the era of non-targeted cytotoxic chemotherapy has fallen greatly down, and the era of personalized, individual, targeted, and immunotherapy is continuing to rise. But there are barriers, of course. Cancer is, as we've just mentioned, a complex disease with potentially a very complex genome. The current panels of trying to look at just small numbers of genes and only the hotspot regions of those genes is extremely limited. We'll see in lung cancer, if all you do is look at EGFR by some sequencing assay and ALK fusion and maybe ROS1 fusion, you leave behind a significant number of patients. that will not benefit from cytotoxic therapy and have targeted therapies just waiting for them, that if you don't sequence and find them, you'll put the patient on the wrong treatment. We have a limitation on the samples that we receive. We do get, of course, resection samples from the surgeons with tumors of large size and the ability to have 70, 80, and 90% tumor cells versus benign, but they're rare. The vast majority of cases we get are limited biopsies, transcutaneous, endobronchial, endoscopic, which may also be small, and we could use the term contaminated by benign DNA from non-tumor cells. And that challenges a system's ability to detect the critical genomic alterations that will define how the patient will be treated. And then finally, we have, and this of course is maybe more significant here in Europe than it is in the USA, the access to the targeted therapies themselves. We may be able to discover targets in European patients, but the ability to get them, the approved in USA targeted therapy may not be, at least at this time, as easy to do here in Europe. And the same holds, of course, for the immunotherapies, which are now also being personalized in a similar way the targeted therapies are. And I'm going to talk about that as I go forward with this presentation. So here's again the highlight. This is formalin-fixed, paraffin-embedded material. These are not fresh samples. The DNA is extracted from processed tissue. We thought it'd be a big challenge when we started doing this, but it turned out we could write algorithms to essentially copy out the DNA alterations caused by processing, by heating, and by embedding and exposure to formalin. This is high accuracy diagnostics. With a pure sample, the ability to detect alterations is relatively easy, but as the impurities occur from benign DNA and the mutant allele frequencies drop below 50%, below 40 and 30 and 20, some assays will continue to find the alterations and others will not. We always have to keep in mind that there is no built-in internal control for this type of work. This isn't fish assays where the wild type means there's only 2 HER2 genes. You know your hybridization worked, you have a built-in internal control. These alterations like RAS mutations and P53 mutations and loss of PTEN and EGFR and fusions with ALK, these are all not normal. These are cancer findings, and we don't have a control in the tissue to make sure our assay works. And I'm going to go into that in detail. So when the mutant allele frequency goes below 20%, a great number of the cancer genome sequencing assays that are currently being done in clinical labs, commercial labs, hospital labs are insensitive and will miss the alteration, especially the deletions. We have testing limitations when we try to do this one at a time. When we try to do immunohistochemistry for part of it and FISH for part of it and then PCR sequencing for another part of it, we run the risk that by the time we get to the assay that will find the critical alteration, we ran out of tissue doing the other assays that didn't find anything. This is why we need an assay that does all of the alteration detection in a single sample. And those alterations I show you know well, the base substitutions, the short insertions and deletions, the copy number changes, which of course include homozygous deletions and gene amplifications, and finally the gene rearrangements and gene fusions. Of these 4 classes, the easiest to find are the substitutions, probably followed by the gene fusions. Amplifications, if your system is working well and you have enough DNA loaded, are relatively easy to find above 10 copies. But between 6 and 10 copies, those amplifications can be difficult to define. And finally, the short insertions and deletions, especially the deletions, are the most challenging to find, and systems that are not sensitive enough, looking for exon 19 deletion in lung cancer in a 20% tumor sample, will miss those in a significant proportion of patients. So false negative, because there isn't a built-in internal control, is the most significant issue with DNA sequencing for precision medicine. Since you don't know if you issue a report with no controls whether your result is a true negative or a false negative, patients are often put on chemotherapy, who have targeted therapy options because the system didn't detect the exon 19 deletion or several of the other important mutations. We need to protect against false negative. It is the single most reason why patients get the wrong treatment who do have a sequencing assay performed. This was done by Foundation Medicine when the test FoundationOne was first developed. We realized that we were going to use the most sensitive platform available at the time, which is of course also the most expensive, and that is the Illumina hybrid capture-based high-seq assay, the reagents of which for one patient can cost anywhere from $600 to $900. Just the reagents, before you have to have a $1 million sequencer and extremely well-trained individuals working from the loading start of the assay to the interpretation and report production of the assay. What Foundation Medicine did was purchase hundreds of cell lines from the HapMap consortium that had the critical 4 classes of alterations that the test needed to detect. So these cell lines had mutations that included substitutions, indels, copy number changes, and gene fusions and rearrangements, and then diluted those cells down to clinical grade and below clinical grade, and run them into the flow cell through the sequencing assay every day, every run, along with the patients. It's not a true negative control. It is a synthetic control, but it allows you to make certain that all of the parameters are in their working order before you start assembling the alterations in each individual patient. And this, of course, was published in December of 2013 in Nature Biotechnology. So the FoundationOne test is a hybrid capture-based Illumina high-seq test customized for formalin-fixed paraffin-embedded samples, simultaneously assesses the 4 classes of alterations, has this unique cell line control system. It has what it believes the highest sensitivity to prevent false negatives and the best pipeline set of algorithms to prevent reporting false positives. It doesn't require a germline sample, but it is capable of determining the germline status more than 98% of the samples that are received, using a proprietary algorithm for determining germline status just published yesterday after a long period of journal selection, shows how we're able to determine germline. But we don't report germline. We're not a germline test. We didn't get a germline sample. We don't have patient permission for germline, and we don't have a genetic counselor ready to receive the results and communicate the results to the patient. So we usually call or email or somehow alert physicians when we detect germline alterations. We do detect them far more frequently than we ever thought we would when we started the company in 2009. And in fact, the last time I checked, we now have 260 Li-Fraumeni syndrome patients, all of which were unknown to either the patient, the patient's family, or the oncologist who submitted the sample when we did the sequencing. We're realizing that germline TP53 mutation is much more common than we ever thought. So that's the test. But more recently, you can see the graphic. The critical points are in not just the upfront, where a pathologist determines the sample adequacy, the amount of DNA that will be extracted, and the at least 20% tumor. If we look at samples that have less than 20%, we start to reduce our ability to detect gene amplification due to over-dilution by benign wild-type copy number DNA in the assay. So we require 20% or higher tumor, and we have to enrich approximately 25% of the cases, maybe a little less, to get up to 20%. But our signature assays are driven by our pipeline, by the algorithms that we wrote to make this test work well in clinical grade material, and those of course are separate for the 4 classes of alterations, and then a report that is customized for the now country where the patient sample came from. US of course is well established, and we're now starting here in Europe to customize reports, particularly to link the right clinical trials to the right alterations. Does no good to have a European patient have their sample linked to clinical trials that are based at the US clinicaltrials.gov. That's not what we're doing, and we're working on this country by country and EU as a whole. Laboratories exist in U.S. in Cambridge, Mass. That's the headquarters of the company. Also in Research Triangle Park, North Carolina. Lab opened in November of last year in Pennsburg, Germany. A lab will open almost any day now in Zurich, Switzerland, and we're expecting by the end of this calendar year to open a laboratory in the Shanghai region in China. The milestones, the company started doing this in early 2010. First major discoveries were the RET alterations in lung cancer, the NTRK alterations in lung cancer, co-discovered the ESR1 mutation as a hormone receptor targeted therapy resistance alteration in breast cancer, BRAF fusions in spitzoid melanomas, FGFR fusions in cholangiocarcinoma, HER2 mutation sequence alterations in lobular breast cancer, and then most recently the tumor mutational load or burden as a way to further customize the immunotherapy. I think you can read through the rest of the release of the genomes. I want to get myself up to speed here to talk a little about lung cancer and beyond schedule. Here are the list of the different assays Foundation Medicine does. The green one I just put in, which is the FoundationOne CDX. This was approved in late November by the US FDA and Centers for Medicare and Medicaid Services simultaneously on the same day, so-called parallel review. It's unique for an assay like this to have both a pre-market approval, not a clearance, but an approval from the FDA, as well as at the same time a decision to give it national coverage by the Medicare portion of CMS. We are launching this quietly and slowly as we complete the comment period and get the go-ahead from the federal government to wide-scale launch it. It's very similar to the FoundationOne test, but it has a slightly larger bait set, more genes being sequenced, and it's done in a slightly different way within a highly regulated laboratory environment. The FoundationOne test still continues, as does FoundationOne Heme, which adds an RNA sequencing component to the DNA sequencing only to increase the sensitivity for gene fusions and rearrangements. This is not a whole transcriptome profile. The RNA is only baited for fusions and rearrangements. We do all of our lymphoma and leukemia samples that way, as well as sarcomas. We're recently going to break it apart and have F1 sarcoma and F1 heme, but right now the sarcomas are done on F1 heme. We launched our blood-based hybrid capture DNA sequencing assay 2 years ago. That's the FACT, F-A-C-T assay, and that's in 2018 going to be slightly expanded in genes in the test, as well as the total mutation burden added to it. So we will have a FACT TMB assay sometime later in 2018 as a commercial test. We also report the microsatellite instability based on our sequencing assay, and that is also just about to be published. How we do that, the TMB technique is published. And then we have our interactive cancer explorer called ICE, which I won't talk about. So here's the FoundationOne test, CDX, in the first column on the right after the list. And let me go through it here in a little more detail. So this test now is approved for all of the lung cancer approved markers. And that, of course, is the EGFR sequence, the ALK fusion, and the ROS1 fusion, the melanoma BRAF V600E, the colorectal cancer KRAS and NRAS, a negative predictor for the anti-EGFR antibody treatments, and the breast cancer HER2 copy number or HER2 amplification, and finally our already approved BRCA1 and 2 somatic sequencing, which of course will include both somatic only and somatic plus germline patients for candidacy to receive the PARP inhibitor rucaparib. There's quite a number of patients who have lung cancer and melanoma and colorectal cancer and breast cancer. So you can see that 34% of US cancer patients might be impacted by the results of FoundationOne CDX. What I want to talk about now is what also do we find when we don't find a companion diagnostic target. So I'm going to move over to lung cancer. and briefly talk about this and finish. The test, of course, detects all the alterations in the lung cancer sample. These are often our most challenging because they're transthoracic fine needle aspiration biopsies or endoscopic endobronchial fine needle aspiration biopsies, and they can be very small samples. We have to be able to detect the ALK fusion, and we find that our technique will detect ALK fusions in cases that the FISH FDA-approved test is negative. In fact, FISH misses 38% of ALK fusions in lung cancer, which is, of course, a very serious problem. For EGFR, the FoundationOne test will detect 18% of false negative exon 19 deletions done by PCR and redirect the patient to EGFR therapy, just like it redirects the patient to ALK therapy. It also, of course, will detect MSI status, which is rare in lung cancer, being MSI high, but it will detect the tumor mutational burden, which for advanced lung cancer can approach as high as 20 to 25% of the patients. But we're now also doing single gene predictors of immunotherapy response combined. At some point, we may have an actual test that's the immunotherapy responsive predictor test based on all of this. We also do PD-L1 IHC testing. And when you go through this list of the approved and directly targeted approaches in lung, which is mainly EGFR, ALK, and ROS1, and then you don't go on further to look for NTRK, for BRAF, for HER2, for MET, for all of these other potential targets, you leave a significant, as you can see, portion of patients who would have received, perhaps in a trial, a targeted therapy, who instead will get second line or third line chemotherapy. And we try to explain this, to promote this, that lung cancer really should start out with a comprehensive genomic profile and shouldn't start out with a triage approach where you look for the low-hanging fruit of EGFR, maybe ALK and ROS1, and then think, well, we didn't find those, what should we do now? This test can be turned around now in about 8 or 9 days in the US. There's no reason to do the one-off, less sensitive, not the entire sequencing portion tests first as a triage when you have a test that can do it all at the highest sensitivity and find all of those alterations in those patients who are wild type for those, and then of course give the immunotherapy guidance as well. So there's a lot of societies now in the US, the National Comprehensive Cancer Network, for example, that are advocating do comprehensive profiling up front, not single gene one-off testing. And this is from their latest publication from the NCCN guidelines. So false negative FISH. This is our 4th patient we ever sequenced. 4th patient, a man from Israel, a water sciences engineer who had a non-smoking background and in his early 40s presented with brain metastases from non-small cell lung cancer. He was sequenced by us after his FISH assay, which you see up here was wild type with no break apart. We found an EML4 ALK fusion on the FoundationOne test. After that, we went stained with the cell signaling antibody to show ALK was overexpressed in his lung cancer. And here are his early responses. This was in early 2011. And now in early 2018, he and his wife have had 2 more children since his brain metastases. He's working full-time, feeling reasonably good on maintenance anti-ALK therapy. So what a difference. If he'd have gone and gotten second-line chemotherapy instead of anti-ALK, he would have died. Instead, he's living a productive life on treatment, not cured, but on maintenance therapy. I mentioned the ALK fusions are missed in 38% of FISH is the only test that's done. Exon 19, as I mentioned, 18% false negative rate in referred samples from patients who had PCR-based sequencing. We resequenced and found the exon 19, and that's a study that is clinical outcome-based. It's not we said it's mutated, they said it wasn't. It's that they said no, we said yes, patient got a TKI and benefited. So it was endpoint proof that we were seeing false negative rates for this critical lung cancer genomic driver. So here's a list of all of the different ones that we've found. We did this study with Memorial Sloan Kettering on their patients. They were using a proteomics-based sequenom sequencing assay. We were using Illumina hybrid capture HiSeq. We reported on all of their negative patients a whole host of alterations. They treated them and got benefit from virtually every one that we changed. The findings were actually so profound that a donor gave Sloan Kettering a substantial amount, tens of millions of dollars, so that they could build an Illumina hybrid capture HiSeq, which of course is now the Sloan Kettering impact assay, just based on this study when Foundation showed this seriously concerned cancer center that they needed to switch to Illumina hybrid capture. This patient was the first patient we discovered the RET fusion in. For pathologists, of course, I always like to teach, you see a papillary architecture in a lung cancer, it's a RET gene fusion-driven tumor until proven elsewhere. If you find a pneumoma body in a lung cancer, it's RET fusion 100% of the time, which then means the patient shouldn't get chemotherapy, although I must admit the RET targeted therapies are not as good as the ALK or the EGFR right now, but there are anti-RET targeted therapies for lung cancer in clinical trials that are apparently doing substantially better in terms of longer disease-free and overall survival than using cabozantinib or the others that are on the market. This is a sarcomatoid lung cancer. This histology gives us an inkling that this will have a MET exon 14 splice site mutation. This was co-discovered by us. It is a different alteration in MET than the MET copy number increase. It's a much more robust clinical assay because it's either got the alteration or not. Where the cutoff should be and calling MET amplification is a very controversial issue. I believe it should be very high, at least 7. Others will say over 5 is amplified. It's driven a lot of failures of anti-MET therapies for lung cancer that should have gotten approved, treating too many patients rather than just the real MET-driven ones. But the exon 14, that's an approvable indication. We get dramatic responses. This patient's mass shrinkage here, this is over a year ago. This patient is doing well. There's no evidence of this disease now on maintenance crizotinib off label, but working well. And here's another example of a MET exon splice site mutation patient who got on the PET-CT substantial response to this. In this case, it was crizotinib rather than the experimental capmatinib. And I just want to finish up. How am I doing time-wise? I didn't check. I'm okay, good. I want to talk about using this type of comprehensive genomic profiling to guide the use of immunotherapy for non-small cell lung cancer. We've been able to now show in multiple different clinical trials using different drugs, both anti-PD-1 like nivolumab and anti-PD-L1 like atezolizumab, that tumor mutational burden is a major predictor of benefit or resistance, and that in a multivariate analysis against most of the other predictors like PD-L1 IHC staining or et cetera, that it is an independent and perhaps the most significant individual marker for predicting benefit. And we use a cutoff of 20 mutations per megabase. We must sequence a minimum of about 1.1 million bases to give an accurate mutational burden. It's not what you think it is. At least it wasn't when I first understood how we were doing this. This does not include any germline mutations or any driver alterations. So if you get a Foundation Medicine report and there's a list of 20 genomic alterations, none of those are being counted for the mutational burden, which confuses oncologists often, saying, how can you say this is low mutational burden? You reported 20 alterations. We're looking at VUSs predominantly VUSs, the kinds of small non-driver alterations that produce peptides that immunize the patient against their cancer. So the goal here is to have a test that predicts the neoantigen load that the tumor is creating to thus predict the availability of a T-cell response if you were to lift the PD-1, PD-L1 blockade that the tumor is currently using to cloak itself from the immune system. I put here a list the different findings. The one table that I want to go into detail because there's been some press releases about this study this week. And this comes from Bristol Myers Squibb, and the drug, of course, is nivolumab. And nivolumab, we did this study with them after they had failed to achieve a frontline indication when compared with pembrolizumab from Merck, which did achieve it using IHC as the required biomarker. If you remember the Pembro approval, about 38% of patients in the frontline stage 4 lung cancer have a PD-L1 greater than 50% of the cells staining. When they treated with pembrolizumab, they got about a 25% patient to benefit with a prolonged survival, and it outperformed frontline chemotherapy. Well, nivolumab from BMS did not get approved. It didn't have the outcome data that was sufficient for approval, whether it was done without a biomarker or when they put their IHC test in. So we went and looked at the tumor mutational burden and reanalyzed the data, and we saw that when there was a high TMB, there was an enormously dramatic difference in overall and progression-free survival when compared to standard of care frontline chemotherapy independent of the TMB. What came out this week was that in the expansion of this with the new data was that the TMB alone without the PD-L1 IHC could predict efficacy in a very significant way when it was used prospectively in the so-called Checkmate 27 trial. So we're expecting that to be submitted to the US FDA to get that frontline approval for nivolumab. In this case, we'll drop down from 38% eligible patients to only 25% eligible patients. But the prediction of long-term durable response, meaning overall survival benefit, is almost guaranteed if the patient has more than 20 mutations per megabase on the FoundationOne assay. Now, there are other individual genes coming out now. We're finding that BRAF mutation in lung cancer is a robust predictor of benefit from the checkpoint inhibitors, and the Mexon 14 splice site mutation. Both of these seem to be associated with a burst of neoantigen production. Most recently, you may have read about the final revelation of why clear cell renal cell carcinoma benefits from checkpoint inhibitors when it does not have a high mutational burden and it may not stain for PD-L1. And the explanation comes with the chromatin remodeling gene PRMB1, which is very near the VHL gene and is impacted in about 40% of clear cell renal cell carcinoma, VHL more like 80%. And it's in the PRMB1 patients with renal cell cancer that's when you see the checkpoint inhibitor benefit. Well, we're seeing this in other tumor types. I just sent an ASCO abstract in this morning on its high frequency in mesothelioma, a disease which we haven't had any treatment for, but now recently we've realized will occasionally have a robust response to checkpoint inhibitor. Well, about 15% of mesotheliomas have genomic alterations in PRMB1, and I think that could be a new application of sequencing for this disease to try to get the right patients on immunotherapy. We've also discovered markers of resistance, the key one being the serine threonine kinase 11, STK11. This is, as you know, a gene when mutated in the germline will give you the Poitz-Jeghers intestinal polyposis syndrome. But STK11, we and 4 other sites, Sloan Kettering, Dana-Farber, MD Anderson, and Bristol Myers, all have the same results on STK11, a robust predictor of resistance to checkpoint inhibitors, even in patients who have other biomarkers that predict benefit. STK11 is not mutated in the somatic in very many cancers at all. It's in the 1% to 2% range in the vast majority. But in non-small cell lung cancer, especially adenocarcinoma, it's more than 15% and can approach 20% in certain cohorts. And we see those patients being completely resistant to benefit. We didn't know why, but since we made these discoveries, and this manuscript is pending, several have come out explaining that STK11 function is required for lymphocyte tracking and for T cells to be able to find targets. And tumor cells with mutations and activating mutations in this tumor suppressor appear to remain cloaked and invisible to the immune system even after you give a checkpoint inhibitor. And then finally, there's the concern of the MDM2 amplification. Only one paper on this from the UC San Diego, the Rosell-Kurzrock group, that described that patients with tumors with MDM2 amplification will undergo hyperprogression if given a checkpoint inhibitor. This, of course, has to be validated. So just real quickly, I'll go through the data and I'll stop. SDK11, this is our cohort. We used the Flatiron collaboration, a clinical outcome company that Foundation Medicine has a contract with and a partnership with to prove the SDK11 prediction. And we used the Flatiron, and this is the Kaplan-Meier for that on a very small cohort of patients. When you add the other sites together, these differences between survival get far more dramatic. Here's the MDM2 from the Kurzrock et al. Paper, or Shumai Kato and Kurzrock et al. And this is where you see this more than doubling of the tumor size within 3 months of the start of checkpoint therapy in patients who have MDM2 amplification. Of course, another group is going to have to validate this before we start considering it a bona fide, validated anti-checkpoint inhibitor biomarker. So here's my final slide. Comprehensive genomic profiling can maximize cancer patient treatment options to both targeted and immunotherapies. It's not just for targeted therapies. When we bring a marriage of the clinical and genomic findings, we can drive a new wave of discovery and great potential to improve outcomes in patients with cancer. And I certainly believe there's no more exciting time than now to be involved in cancer genomics and translational research. Thank you very much.